Introduction. Welcome to today's lecture on cell cycle regulation and its implications in various biological processes. In this comprehensive script, we will delve into the intricate molecular mechanisms that govern the cell cycle, the critical checkpoints that ensure its fidelity, and the consequences of dysregulation in diseases such as cancer. Additionally, we will explore the role of signaling pathways, DNA damage repair mechanisms, epigenetic regulation, and development in maintaining precise control over cell growth. So without further ado, let us embark on this fascinating journey into the world of cell cycle regulation. The cell cycle is a highly regulated and coordinated series of events that enables cells to grow and divide. It consists of distinct phases, including the G1, Gap1, S, synthesis, G2, Gap2, and M, mitotic, phases. During G1 phase, cells prepare for DNA replication by synthesizing proteins and increasing their overall size. The S phase is marked by DNA replication, where every chromosome duplicates itself. G2 phase follows DNA replication, during which cells continue to grow and synthesize proteins necessary for cell division. Finally, the M phase encompasses both mitosis, the division of the cell's nucleus, and cytokinesis, the division of the cytoplasm, resulting in two identical daughter cells. The cell cycle is precisely regulated by a variety of molecular players, including cyclins and cyclin-dependent kinases, CDKs. Cyclins are proteins that accumulate during specific cell cycle phases and bind to CDKs, activating them. Once activated, CDKs phosphorylate target proteins, initiating the necessary events for cell cycle progression. CDK inhibitors also play a crucial role in controlling the cell cycle by binding to cyclin CDK complexes, preventing their activation and thereby maintaining cell cycle arrest. To ensure accurate progression through the cell cycle, several checkpoints act as quality control mechanisms. The G1 checkpoint, also known as the restriction point, determines whether a cell should continue with the cell cycle or enter a non-dividing state called the G0 phase. The G2 checkpoint assesses DNA replication fidelity, while the M checkpoint ensures proper attachment of chromosomes to the spindle apparatus before entering anaphase. Failure to pass these checkpoints can lead to cell cycle arrest, DNA damage accumulation, and potential genomic instability. Cell growth is intricately regulated by external signals, including growth factors and hormones, which activate signaling pathways that ultimately lead to cell cycle progression. Receptor tyrosine kinases, RTKs, and their downstream effectors, such as the Ras-Raf-MOPK and PEEK-ACT pathways, play pivotal roles in transducing these signals into intracellular responses. Dysregulation of these pathways can contribute to uncontrolled cell growth and tumor development. Within the cell, various signaling cascades ensure proper coordination of cell cycle events. The well-studied cell cycle control system, known as the mitotic cell cycle oscillator, involves the sequential and cyclic activation of cyclin CDK complexes, culminating in the entry into mitosis. Additionally, the anaphase promoting complex, APC/C, regulates the degradation of key cell cycle regulators, ensuring the timely progression from one phase to another. Tumor suppressor genes, including P53 and PRB, play vital roles in maintaining cell cycle integrity. P53 acts as the guardian of the genome by monitoring DNA damage and promoting cell cycle arrest to allow DNA repair. PRB controls G1 to S progression by inhibiting E2F transcription factors, preventing premature DNA replication. Loss or mutation of these tumor suppressor genes can disrupt cell cycle regulation and contribute to tumor genesis. Cell cycle dysregulation can be attributed to various factors. Some of the common mechanisms include mutations in oncogenes, leading to constitutive activation of cell cycle promoting proteins, and loss or inactivation of tumor suppressor genes. Additionally, aberrant regulation of cyclins, CDKs, and their inhibitors can disrupt the delicate balance of cell cycle progression. Environmental factors, such as radiation and certain chemicals, can also induce DNA damage, driving cell cycle dysregulation. Apoptosis, or programmed cell death, is a tightly controlled process that eliminates unwanted or damaged cells. It plays a critical role in maintaining tissue homeostasis and preventing the accumulation of aberrant cells. Apoptosis is regulated by a complex interplay between pro-apoptotic and anti-apoptotic proteins, ensuring a delicate balance that determines cell fate. Dysregulation of apoptotic signaling can contribute to various diseases, including cancer and neurodegenerative disorders. Cells have intricate mechanisms to repair DNA damage and maintain genomic integrity. These repair pathways include base excision repair, BER, nucleotide excision repair, NER, and DNA double-strand break repair, DSBR, among others. These processes involve a series of coordinated steps, including damage recognition, recruitment of repair factors, and DNA synthesis or recombination. Failure of these repair mechanisms can result in the accumulation of mutations and genomic instability, predisposing cells to cancer development. Defects in DNA repair pathways can lead to genomic instability, a hallmark of cancer. Individuals with inherited mutations in DNA repair genes, such as those associated with hereditary breast and ovarian cancer, BRCA1/2, are at increased risk of developing cancer due to impaired repair of DNA damage. Furthermore, acquired mutations or epigenetic alterations in DNA repair genes can contribute to the accumulation of genetic aberrations, driving tumor initiation and progression. Epigenetic modifications, including DNA methylation, histone modifications, and non-coding RNA molecules, play pivotal roles in regulating gene expression and cell growth. These modifications can modulate chromatin structure and accessibility, influencing the binding of transcription factors and other regulatory proteins. Dysregulation of epigenetic marks can alter gene expression profiles, disrupt normal cell cycle control, and lead to abnormal cell growth observed in various diseases, including cancer. During development, precise control of cell growth is crucial for the formation of complex structures and tissues. Developmental signaling pathways, such as Notch, Hedgehog, and WNT, play pivotal roles in orchestrating cell proliferation and differentiation processes. Spatial and temporal regulation of these pathways ensures proper tissue growth, while dysregulation can result in developmental abnormalities or cancer initiation. In conclusion, the cell cycle regulation is a complex and highly orchestrated process, involving numerous molecular players, signaling pathways, and checkpoints. Maintaining cell cycle fidelity is essential for normal development, tissue homeostasis, and prevention of diseases such as cancer. Dysregulation of cell cycle control mechanisms can result in uncontrolled cell growth, genomic instability, and increased predisposition to cancer. Understanding the intricate details of cell cycle regulation provides valuable insights into potential therapeutic targets for cancer treatment and opens avenues for further research in this fascinating field.